For example, the cell which is rapidly dividing, an epithelial cell, your NCERT says may have a life cycle of around 24 hours. The yeast is again a eukaryotic cell, your NCERT says it would be around 90 minutes. So these prokaryote cell actually divides much faster. So we are looking at some cells which are continuously dividing, and these cells, they will actually typically directly enter into the S and so on and so forth. But we are looking at some cells which are terminally differentiated, which are senescent, and which may divide. They are not yet dividing, but may divide when needed, when called upon to do so. So we are looking at some cells which are not very regularly dividing, but they can divide. They have this potential, and they do divide whenever they are called upon to do so. And we will place the liver cell in this particular example. All these type of cells now just listen it very carefully what I am saying. All these type of cells, they exit the cell cycle from G1. They exit the cell cycle and suspend the events of the cell cycle. So they exit from the G1 phase, suspend the sequence of the events of the cell cycle. Again, understand this. They exit from G1 and they land up in a phase which is called as the G0 phase. Now this G0 is incidentally called as quiescent, but there is nothing quiescent about it because it is a still differentiated cell. It is undergoing all the functions, it is performing all the functions that it is required to, and therefore the cell is metabolically active. It is not an metabolically inactive state. Please remember this. The cell is carrying out a lot of metabolism, but the cell has withdrawn itself from the cell cycle, has exited in the G1 phase, landed up in what is called as an extension of G1 phase, which is called as the G0. It is not requiring to divide right now. It may be this cell, it may be this cell, or it may be this cell. So that means this withdrawal can be, you can easily appreciate, can be temporary or it can be permanent. So withdrawal from the cell cycle, that means exiting what is called as the G1 phase and landing up in the G0 phase, can be temporary or permanent. This should be very easy, and therefore you do the question that a neuron is a cell in the human body that normally doesn't divide ever. I hope this is absolutely clear. But what about the liver cells? They have temporarily withdrawn themselves, and they have suspended the events of the cell cycle for the time being. This type of here the suspension is permanent, here the suspension is temporary. Now what is important to understand is that, okay, now let us look at this cell, the cell that may actually divide and is now called upon to do so. Now this cell has exited from the G0 phase, and if it is required to divide again, it will re-enter the G1 phase. And it will re-enter the G1 phase because it cannot directly go into the S phase without crossing this checkpoint which is present near the end of the G1 and just before the S phase. It has to cross this checkpoint. I hope this is absolutely clear. It has to cross this checkpoint, and then it will be able to enter into the S phase where the cell actually synthesizes the DNA, replicates the DNA. So for example, when entering into the S phase, a cell has 2n content of DNA. This content will gradually increase during the cell cycle, during the S phase, and by the end of the S phase it becomes 4n. So please understand, the cell is replicating its DNA, so it is taking its time. So just entering into the S phase doesn't make it a 4n DNA content. The 4n would be at the end of the S phase. I hope this thing is absolutely clear. Now we know that the cell has replicated its DNA and now is committed to divide. Now it will complete the events of the cell cycle. So which